bioengineering, one of the co-founders of the department at Stanford University, and he is also an HHMI investigator. Stephen's contribution to the development of new biotechnology at the interface between physics and biology have been widely recognized. And previous to moving to Stanford, he was at Caltech, where he pioneered research programs on single molecule biophysics and soon expanded to include fundamental inventions such as single molecule sequencing and microfluidic large-scale integration and their application to biology and human health. Interesting for all the younger generation that usually is the group that comes to DREAM, his career was very colorful in that he started doing mathematics and went to IBM Research to do some internships where he met fractal guru Benoit Mandelbrot, where he sat in a room with someone who eventually was his advisor at Oxford, and went to Caltech, where he also, by interactions, started to think about microfluidics, and his career built up from there. So it was the little perturbations that make big impacts in careers of scientists. Of his many honors, I will mention the MIT Lemelson Prize, the Royal Society of Chemistry Pioneer of Miniaturization Award, and the NIH Director's Pioneer Award, but he has many others. He is elected Fellow of the National Academy of Inventors, the National Academy of Sciences, the National Academy of Engineering, and the Institute of Medicine. And today is Dreams and Systems Biology keynote speaker.
Well, thank you so much, Gustavo, and the rest of the organizers for the opportunity to come here and speak today. I'm going to tell you about some work we've been doing in single-cell genomics in the lab over actually quite a number of years. And this is a very hot area at the moment. It's getting a lot of press. NIH has got programs funding it and whatnot, and sort of papers coming out every other week, it seems. And so I like to remind people always at the beginning of the talk that it's actually a very old field. And in fact, it dates back to the 19th century. So Walter Fleming noticed that in some cells and some insects, there was a phenomenon that enabled you to see the chromosomes under an optical microscope. And that phenomenon is called polytene chromosomes and is due to sort of a huge polyploidy that happens there. And it was more than just a curiosity. It was put to practical use probably for the first time in the 1930s at Caltech by Calvin Bridges, who used it as a method to map out the Drosophila genome at low resolution. And so he used the salivary glands where this happens and was able to map the genome. And that sort of mapping technique was used for quite some years in the Drosophila community to track differences between different lines and individuals and species and so forth. You know, it remained, because it depended on that sort of obscure biological effect, sort of a niche thing. And I think, you know, what has really brought it to the fore and given the field so much momentum right now is the confluence of three major technological advances. So one of them is the development of biochemical amplifiers like PCR and MDA, also known as multiple displacement amplification. And PCR, of course, was the first biochemical amplifier invented in the 1980s. And almost from the very first paper, people realized that such a high-gain amplifier ought to be useful for looking at gene expression and gene sequence from single cells. And there's a literature that dates all the way back to that time. But again, it remains sort of a very small field because the other two pieces weren't there that ended up being really enabling. And so one of those pieces was the development of high-throughput sequencing technologies that lets you look at gene expression and genome sequence at very low cost and incredible depth and resolution, which you're all familiar with. And the third one, which is also important, is the development of ways to manipulate the single cells and put them where you want, which lets you do these measurements at large scales on large numbers of cells. And so examples of these are fluorescence-activated cell sorting and microfluidics and so forth and so on. When you have all three of these technological pieces, all of a sudden you have a very general and powerful tool to look at virtually any complex tissue or heterogeneous biological system, whether it's a microbial ecosystem or a human organ. And so it was named the Method of the Year by Nature Methods. And then if you look at the January 2014 issue of that journal, a number of us wrote sort of perspective pieces about where we think the field is going. And that's, I think, a reasonable introduction to the field if you're interested beyond what I'm going to talk about today. So one of the ways I got pulled into this field was the work that we did developing microfluidic technologies in my group over a period of a decade. We thought about this as sort of the integrated circuit of biology and a way to automate biological measurement in the same way that the integrated circuit automates computation. And for that sort of automation, you don't need gates and transistors, you need plumbing, pipes, valves, pumps, and so forth. And we worked out ways to build chips that had tens to hundreds of thousands of micromechanical valves on them, and use them to explore a variety of problems in biology. That's well beyond single-cell genomics. Many other fun things to do with this plumbing. And this technology that we developed formed the basis of Fluidigm Corporation, of which I'm a founder, and in the interest of full disclosure, I'll tell you that right now. And through the talk, I'll try and point out where we used homemade devices and where we used their sort of commercial descendants. And so we thought these plumbing tools would be a really useful way to prepare unique materials for genomic analysis, especially in the context of microbial genomics, where, as you might be aware, only maybe 1% of the microbial universe can be grown in laboratory culture, and so 99% of the diversity out there can't be accessed by conventional means, and in particular, it's very difficult to understand the genetics. And so we thought, wouldn't it be nice if we could build a device that allow us to isolate individual microbes, amplify their genomes, and then take that amplified material off the chip and sequence it or analyze it by whatever method you liked. And we did that and explored a number of really sort of fun problems in microbial ecology, did a sort of ever more sophisticated set of genomes from single cells. I'm not going to dwell on it. These are a few of the publications right here. But in doing this work, we started to think about what are the sort of technical aspects of doing a single cell amplification, or more specifically, a single molecule amplification, starting from one copy of the genome. If you want to amplify, how well can you do? And this led us into a pretty interesting excursion into sort of the details of these amplifiers. And the one we like to use, we just sort of as our first attempt, was this one called multiple displacement amplification that was developed by Roger Laskin. It's got a lot of nice properties in that it's isothermal, it's got enormous gain, it doesn't require any a priori knowledge of the molecules you're trying to amplify. Those are all good properties. And as we started to play with this, actually, let me just tell you a bit about how we do the experiments. So the basic unit structure on the cell, on the chip, looks like this. It's a series of chambers connected by valves, and you, by fluidic manipulation, trap a cell in the first chamber, and then you fill it by opening the valve, by pushing in a new reagent there, and then allow them to mix by diffusion. And then you can open the next valve, push in a new reagent, and allow them to mix, and so forth. And in this way, you can do very complex sort of manipulations of the material there. So, for example, lyse the cell, denature the DNA, neutralize, put in the amplifier, do the amplification, and then recover the material. And this is what the chip looked like. There were 8 such unit cells and a control. And as we started to play around with this using E. coli as our practice organism, we got in touch with Roger, and it turned out that he had a postdoc who was trying to do the same sort of thing, single cell amplification, but in microliter volumes. And so we decided to team up and do a comparison of how the amplifier performed in nanoliter volumes and in microliter volumes. And so we did a bunch of single cell, my postdoc, Jan Marcy, did a bunch of single cell E. coli genome amplifications and sent the amplified lysates down to Roger's postdoc, whose name was Thomas Ishui, and Thomas did a bunch of microliter amplifications by micromanipulating the cells with a pipette, much more painful. And then Thomas did sort of the side-by-side quality control on both samples. And what he did was choose a bunch of genes across the genome and do quantitative PCR to see the relative representation. And each colored line here is a different single cell. And what you can see is that in the smaller volumes, you're getting much more uniform amplification with many fewer dropouts and better representation of the genome. And that turns out to be a sort of generic effect across most of the biochemical amplifiers we've looked at, including PCR. If you're trying to amplify from single molecules, as you shrink the volume, the performance improves quite dramatically. And it has to do with some of the details of the physical chemistry, how these things work. It's sort of interesting. I'm happy to chat about in the questions. I'll just say that as this field gained momentum and there's sort of more creativity in the development of a number of other amplifiers, we decided to go back and revisit some of those measurements recently, and in particular to compare MDA with the Rubicon amplifier and the Malbec, which are quite similar sort of hybrid type amplifiers that use both isothermal and PCR along the way. And we sort of published those results last year, and there's a very nuanced story to it all, but let me just sort of point out two aspects of it that convinced us to stick with MDA, which we happened to pick by accident as our first amplifier. It turns out to be a good one. One is the tremendous specificity that you get, and that's really a function of the volume again, so it reduces contaminants to shrink the volume. As you look at a single cell done in microliter volumes, either with Malbec or MDA, so that's MDA, that's Malbec, that's Rubicon, the vast majority of the stuff is not mapping properly because it's from other organisms, and the green stuff, which is what you're trying to, E. coli you're trying to amplify, is a tiny fraction of what's there. Whereas if you do a single cell in microfluidic volumes, the specificity is enormously higher. And it approaches that of what you get using bulk experiments. So that's and the fact that we can do the MDA in small volumes ends up being an important corollary to that. Second is the error rate of these amplifiers, which if you go and measure sort of the substitution error rate, it turns out that they're basically limited by the enzymes, the polymerases you're using for the amplifier, and the polymerase used for MDA is just much, much better, and sort of almost an order of magnitude lower error rate. And so that's an important issue for many of the questions we're interested in. Okay, so we had sort of been doing all this in the microbial universe because it was cost effective to do the sequencing. But of course, the fact that sequencing costs were decreasing very dramatically during that period meant we just had to wait a few years, and all of a sudden it became feasible to think about doing single cell analysis of eukaryotes, in particular of humans and mice. And you may ask, why would you want to do that? It turns out that not only do you have genomes that are different from each other at the average level, each of your cells have different genomes for different reasons. And some of these reasons and some of these cells have been known for decades. So one example is the immune system, where a B cell that's decided to make a particular antibody makes such a strong commitment to that antibody that it will reprogram its genome and cut and splice and sort of hardwire that antibody sequence into its genome, and there's no going back. It'll never change its mind and make anything other than a descendant of that antibody. Sort of like Cortez burning his boats when he got to the New World. So if you look at a personal genome and look at the immunoglobulin heavy chain, antibody heavy chain, you'll see that it's just a mess in anyone's personal genome because it's averaging many different cells that have different genes there. Another example are gametes, sperm and egg cells. And they're different from the other cells in the body, not only because they're haploid instead of diploid, but also because they've been through the process of meiotic recombination. And so there's been a bunch of cutting and splicing of the genetic material you inherited from your mom and your dad to create a bunch of new, potentially novel offspring in terms of what their inheritances from their grandparents. We spent quite a bit of time thinking about that, and I won't talk about that today, but I'll mention it at the end. And finally, I'll point to cancer as kind of the limiting state of aging and cell division. So every time a cell divides, there's a few replication errors. And if you're unlucky in where those errors happen, you'll get cells that will proliferate without any restraint, and then they'll form a tumor and become a cancer. And the tumor itself is genomically heterogeneous. And so it's, of course, of profound interest to understand that sort of variation as well. And so we've looked at all of these in different ways. And I'm just going to take a few minutes to tell you the one aspect of dissection of cancer heterogeneity that we worked on recently. This is the work of Chuck Gawad, who was a clinical fellow in the lab and recently left to start his own group at St. Jude. And for that work, Chuck began to use Fluidigm C1 chip, which is the commercial version of the chip we had developed in the lab to do the genomic sperm analysis. And this is a chip that allows you to trap almost 100 cells on it, lyse them, amplify the genome in a very similar unit cell to the one I showed you for our microbial work. In the case of humans who are diploid in most of their cells, if you ask what's sort of the limiting error or the largest error in trying to characterize a single cell genome, it ends up being an allele dropout rate, which is to say heterozygous sites in the genome in which there's some bias and you lose track of one allele and they appear falsely homozygous. And you can get a sense for how that works by looking at this raw data here. Well, these are from a HapMap cell line that's been extensively genotyped. And each row here is a different single cell whose genome has been amplified. And we've then queried the genotype at a particular location at a SNP in the genome, 2 per chromosome, each column being a different one, so marching across. And the bulk DNA result is shown down here. It's all been color-coded so that blue is heterozygous and red and green are the 2 homozygous alleles. And so anywhere there's blue down here, you'll want to check up here to see if it's also blue, and if it appears to be red or green, that's a false result, a falsely homozygous result, because one allele dropped out and wasn't measured properly. And if it's white, then you've had a total dropout and you haven't made any measurement at that site. And you can see that there is some rate of allele dropout, and trust me, that ends up being the dominating thing. If you start doing statistics on this and ask what does it look like, it's something like this. So about half the cells have an allele dropout rate of less than 10%. About another third of the cells have an allele dropout rate between 10 and 20%, and the remainder have higher allele dropout rates. And the question is, well, what does that mean? Is that sufficient for making any kind of interesting measurement, or is it too high to be useful? And I'm going to argue that it's actually perfectly adequate for asking a number of interesting questions about cancer biology, as long as you're looking at enough different SNPs in this cell, at enough different loci. So keep that thought in mind and those numbers, and let me now tell you about the particular problem that we worked on, which is ALL, acute lymphoblastic leukemia. This is the most common pediatric cancer. This is what Chuck treated as a physician. There's several thousand cases a year in the US. Their cure rate is pretty good, about 80% of the kids get cured. But the ones who don't, there's often a recurrence, and that's a problem. And this is known to be a polyclonal disease, and people have measured that by looking at sort of bulk measurements and to try to estimate the dominant clone in a given tumor. And when people do that, they can measure what the clone is at diagnosis, and for the kids that relapse, they can do the measurement again and see if it's the same dominant clone or not. It's just asking what's the biggest piece of the tumor there. And only 8% of the time is it the same clone at relapse as it was at diagnosis. So these things are changing. Other clones are either evolving or taking over. And so about a third of the time, it's clearly the dominant clone at relapse is some evolution from the diagnostic clone, and you can see that there's a relationship. But half the time it's come from some ancestral clone that's quite different, and a small percentage of the time it's a completely different disease. So we thought it would be very useful to try to measure the entire clonal structure of the tumor at one time point, in particular at diagnosis. And if we could develop ways to do that, we thought that that might stimulate ways to think about treating all the clones at once, and maybe that will improve outcomes overall. And so the way we did that was to look at a bunch of patient samples. Chuck did bulk exome sequencing of tumor and normal to identify putative variants in the tumor. He then developed panels for each of those variants and validated them using a microfluidic tool from Fluidigm called the Axis Array to confirm that. And then for each tumor, these are liquid tumors now, so you can take the bone marrow sample, load it into the C1 chip, do the amplification of the single cell genomes, pull the lysates out. And now it would be still cost prohibitive to do whole genome sequencing on large numbers of these cells. So instead he used his panels where he had found what are the interesting places for each patient, and did it anywhere from a dozen to several dozen SNP loci from each single cell. And from that, was able to analyze the clonal architecture of the tumor. And let me show you a few of the things we learned from that. So if you look at the bulk data and sort of histogram allele frequencies, it looks something like this. So this is a violin plot, which is a histogram turned on its side and given this reflection to look aesthetically pleasing. And in a sample that's a pure genome, there's only one genome in it, or single clone, you'd expect to see allele frequencies of only 50% and 0%. These are the minor allele frequencies. So at homozygous sites, it'd be 0, at heterozygous, 50%. But you can see there's a bunch of non-trivial allele frequencies here, which says you have a mixture of genomes. And so there's some mixture of clones here. And people have tried to infer the clonal structure from such distributions. It's just something that, as I hope will convince you, is doomed to failure. It's something that will never succeed because, as a long story short, what turns out to happen is it's not unusual to find two clones that appear at roughly the same frequency, which you can then never separate out from those bulk results. And so here's what the single cell data looks like from this particular patient sample. Each row is a different single cell here. Each column is a different SNP that's been interrogated. And white means wild type, and red means mutated. And you can see that as these are clustered together, there are two clones of roughly equal size. And they're quite distinct. So there's a bunch of mutations that happen in one and not the other, shown here, and a small number that are in both. So those are sort of the most ancestral mutations right there that go back to their common ancestor. And you can start to build a little tree that looks something like this. There's some, they share a common ancestor, that's clear from these two, and they've evolved in these two different clones that are roughly equal size. Now, coming back to the question of dropout rates, you can see that no single cell sort of perfectly recapitulates the clone, and there's sites of dropout here. But the point is, if you measure enough sites within the clone, you can overcome that dropout rate. And as you can imagine, there's a way to make that statement very precise mathematically, and to make estimates of how many SNPs per clone one needs to measure in order to convince yourself that you're not looking at numerical artifact. And we looked at that in a very detailed way, and I'm not going to dwell on it here, but I'll refer you to the paper and the supplement. But let me show you a little bit more data. Here's another patient. The bulk allele frequencies of the tumor, again, sort of a blob. There's something happening that's non-trivial, but you can't tell what it is. In this case, the clonal structure looks like this. There's now 3 clones, 1, 2, 3, 2 of which are very closely related, and the third of which is more distant, but they all share a common ancestor, okay? And so the tree now, look at the phylogeny, looks like this, okay? There's some common ancestor. This one is the more different one, and these two are share a more recent common ancestor. And so we went and did this for half a dozen patient samples, more than 1,000 cells, about 1,400 cells in total, and this sort of summarizes what they look like. That's Chuck right there, who led the work, and this is Winston Coe, a student who also helped out on the project. I think I won't dwell too much on more of the details about this, but I'll just point out a couple things that popped out from doing this as we built the phylogenies. One of which is that in several patients, we saw KRAS emerge as a late mutation. So there's one right there. So KRAS only emerges as a mutation here, and it's not an ancestral mutation, it's a recent one. And so in contrast to solid tumors, where it's viewed as a driver and an originator, in these liquid tumors, it can emerge quite late in the process, and which also tells you there must be some interesting compensatory mutation or related pathway here that allows these other clones to keep up and compete. This I view as just sort of a beginning. I think we've established a measurement technique that allows us to say something quantitative and rigorous about the clonal structure of these tumors based on these single cell measurements. And I think going forward, it's going to allow one to make a sort of very deep connection between these ideas of evolutionary biology and cancer, which have been discussed for quite some time in the literature, but only in the most qualitative ways. This will allow them to become sort of rigorously tested and investigated. And I now want to change gears and for the remainder of the talk, move from genome analysis to transcriptome analysis at the single cell level. And this is something we've been interested in for quite some time and developed sort of a succession of devices to do single cell isolation, cDNA preparation, and gene expression analysis. And we had kind of been puttering around with ways to do whole transcriptome analysis as well. Others began to publish that before we did because we were not satisfied with the quality of our data. But, as it turned out, people thought it'd be interesting just to say, we'll publish that we've got presence or absence of genes from single cells. And so then that spurred us to ask the question, well, can you go beyond that and ask questions relating to quantitative gene expression levels in single cells based on whole transcriptome analysis? And we had done these many years of work using PCR-based methods and gotten to understand the errors and the technical issues of those measurements quite well, and convinced ourselves that we could do quantitative measurements with PCR, and thought we could use that as a baseline for these single-cell RNA-seq methods. And so we did some experiments where we compared a bunch of tube-based sort of large volume transcriptome amplification methods with Fluidigm C1-based system and did all sorts of comparisons, whole transcriptome against whole transcriptome, different amplification chemistries, PCR versus RNA-seq. We published it in Nature Methods in 2013. This is the work of Angela Wu, whose picture I'll show you in a moment. But I just want to point out a few sort of take-home messages that came out of that. And the first has to do with just reproducibility from cell to cell. And this is not in a quantitative way. This is just, is the transcript there or not? Let's forget about expression levels, just presence or absence. And with qPCR, it's sort of 90% level, whether you do it in large or small volumes. It's similar. You can see you can get much worse performance generally when you're doing whole transcriptome RNA-seq from single cells. But it turns out the best performing one is the one done in small volumes. And you take the same chemistry, do it in a large volume, a small volume. It's performing better in the smaller volumes. This relates back again to these interesting effects of how the amplifiers behave when you shrink the volumes. Second of all, I want to try to convince you that you can actually make quantitative measurements here. And so that's Angela right there, who's just starting a faculty position at Hong Kong UST in another month. What we did in each of these experiments was to include spike-in control molecules that are NIS-traceable RNA standards, the so-called ERCC control set. And so every chamber not only has a single cell in it, which we lyse and measure the transcriptome, but it's also got these control molecules, which are very nice because there's a large number of different RNA species in each tube, or each control set, which appear at different amounts, and you're given a list from NIST of each species and how many molecules of each species are there in the tube. And so you can do a measurement where your x-axis here is number of molecules per experiment in absolute units of molecules. We know that. And after we do the amplification sequencing, we get out a quantity, which is not so easy to think about. It's called FPKM or RPKM, reads per kilobase map. It's sort of a standard unit of RNA-seq. And it's never been something that's been very intuitive to me about how to think about that unit. But it turns out there's a nice back of the envelope calculation you can do that makes FPKM and RPKM an intuitive unit for single cell measurements. So if you assume that there's a few 100,000 mRNA transcripts per cell, you can then work out that 1 FPKM is about 1 molecule. So in single cell experiments, the interpretation of FPKM or RPKM is about molecule. And you can see that that more or less works here, okay? So this is a log scales on both axes, and so 1 molecule in and there, right about 1 molecule or 1 FPKM out. It sort of works. As such, you should think about what it means to have FPKMs of less than 1 in such an experiment. It's less than 1 molecule per cell, which will only make sense in an average interpretation where you're averaging many cells together. And that's, in fact, what this graph shows here. Each point is a different RNA species, average across 96 experiments, different single cell measurements in the chip. And you can see that when we're putting in molecules at less than 1 per chamber, we're also measuring them out at less than 1 per chamber, but only by averaging across many chambers. Finally, you can ask about the sort of accuracy of this sort of measurement. And as I mentioned, we had become very experienced and confident in what it meant to measure gene expression by PCR from single cells. And so we thought, okay, we'll look at the same genes by single cell qPCR and by single cell RNA-seq and ask, do they agree in a quantitative way? And it turns out they do, both in the large and small volumes. As you average across each of these points is a different gene, average across many cells, and you can see that the slope of the line is very close to 1 and the correlations are very high. And so when you're averaging gene expression across many cells, you're getting reasonable results. Where does the rub come in all this? Well, the rub comes in looking at a given single cell and trying to extrapolate from that. And for that, you want to go deeper than the average and look at the distribution of gene expression values. This is an example of that. So this is now comparing RNA-seq in red and qPCR in blue on single cells on a gene-by-gene basis, and looking at a histogram of the distributions across the single cells. And you can see that for some genes, they're quite comparable. So if you look at the widths of the distribution, we are confident that in most cases, the widths of the qPCR distribution represent the true biological variation and not the technical artifacts of the measurement. And you can see how the RNA-seq does. Here it's pretty good, but in many other cases, the RNA-seq is quite broader than the qPCR distribution, indicating that there's a substantial amount of technical noise there in the RNA-seq measurement that is introduced by the amplification, and one needs to average across many cells to get an accurate estimate of the true value of that gene in a given cell type. So that's a caveat for you to bear in mind. We have then gone on to use this approach to look at heterogeneity and cellular hierarchies in a number of different complex tissues in both mouse and human. And I'll tell you now about one we've done in collaboration with Mark Krasnow to look at the developing lung in mouse. This is work that was led in my lab by Barbara Treutlein. And we collaborated with Doug Brownfield and Tushar Desai in Mark's lab on this. And we were interested in looking at something that happens in lung development called sacculation, which is how the alveoli form. And it has sort of been known that there were 4 cell types associated with a lung, and 2 of them are alveolar cell types. And so one of them is the big flat cells. It gives all the surface area where the gas exchange. It's called alveolar type 1. The other one is very creatively called alveolar type 2. It's these little cuboids that secrete things to feed the type 1 cells. And then there's 2 cells from the bronchiole that were known, the Claris cell and the ciliated cell. And these cells were sort of defined by a small number of genes. So the genes are shown here in the different colors. In some cases, only 1 or 2 genes known per cell. There's not a lot known, and that was kind of the operational definition of the cell types. And what we wanted to do is ask a few questions. 1, are there other cell types that have been missed up to this point? And 2, what are the other genes that are sort of uniquely expressed by these different cell types? And so the experiments were to take either embryonic mice at days 14, 16, or 18, or adult mice, separate the lungs, dissociate the cells from the lungs into a single cell suspension, do some gentle purification of epithelial cells, and then load the cells into the C1 chip, where we would either do whole transcriptome amplification or targeted qPCR amplification of a set of 96 genes, which we could then read out either on RNA-seq or by qPCR. We found it was very effective to use these qPCR measurements as ways to test what was coming out of the very high dimensional RNA-seq measurements. And there's a lot of interesting biology here. Let me just give you a flavor of a few of the highlights that came out of this. So this sort of shows, this axis is average gene expression. This is the CV, or the variation in gene expression across many different single cells. We looked at a few hundred cells in this experiment. Each point here, data point, is a different gene. And so you're looking at variation as a function of how strongly the gene is expressed. And the vast majority of genes are in this big cloud that, the more highly expressed the gene, the lower the CV, as you'd expect from counting statistics and whatnot. But there's a bunch of genes here that are outliers. They're all been colored blue. So these have a very high coefficient of variation with respect to their expression level. And it turns out the reason for that is that these are genes that are expressed uniquely in one cell type and not in others. And so if they're on in some, off in others, you can imagine that creates a huge spike in their CVs. And as you do unbiased analysis, those are the ones that always pop out as the ones that create different cell types, those ones of high CV with respect to gene expression level. And so using those genes, you can sort of cluster the cells together. Each row is a different cell here. Each column is a different gene. And when they're clustered together, you can see there's very clearly structure emerging, okay? And so each different sort of gene expression block indicates a different putative cell type. And if you count, you'll see there's 1, 2, 3, 4, 5 different cell types emerging from this. Already, it appears that we've stumbled onto a new cell type that hadn't been recognized. And we began to give names to these different cell types because of the genes they were expressing. So you might remember FOXJ1 was the gene that defined the ciliated cell type. It's basically only expressed in this group here. So we call those must be the ciliated cell types. Likewise, SCGB1A is the gene that defined the Clara cells. It's only expressed in these guys. A little leakage into there, but this sort of defines the Clara cells. Likewise for AT1 and AT2. Okay, so these are the AT1, these are the AT2, and you can see that this fifth group down here is expressing genes that are differentiation genes related to both AT1 and AT2. It turns out that this is what's called a bipotent progenitor. So it's a progenitor cell that hasn't decided what it wants to be when it grows up yet, AT1 or AT2, and it expresses both sets of genes, then eventually turns one set off. And you can see that from the qPCR data as a function of the age of the mouse during development. So here's a loading plot in the PCA, principal component analysis. You can see that at day 18, the AT1 genes and the AT2 genes are all pointing in different directions, indicating two different resolved cell types. But if you do the same experiment on a mouse from embryonic day 16, 2 days earlier, you can see that the cell type is not resolved. They're all pointing in the same direction, okay? And that's because it's predominantly composed of these bipotent progenitors who, as the mouse ages, they're going to choose identities, fates in one direction or the other. And you can sort of get more deeply into the story, identify more genes, but basically it sort of looks like this. As the lung is developing, the alveoli, there's a progenitor cell whose descendants will either be AT1 or AT2, and we can identify the genes that are involved in that through these measurements. Okay, how much time do I have left? 5 minutes. Do you want me to stop now and take questions or tell you another little vignette? A vignette, okay. No hesitation. So let me tell you about a similar story now, going to human tissue and going to human brain. This was done in collaboration with Ben Barres and Melanie Hayden-Gephardt. So Ben's a neuroscientist, Melanie is a neurosurgeon, and the work was done by Spiros Darmanis in my lab and Steve Sloan in Ben's lab. And we were sort of in a unique position, thanks to Melanie, of being able to obtain adult human brain tissue from living donors. Because one of the procedures she was doing was to treat patients with epilepsy. And she'd go in and remove the epileptic center. But to get there, and that sort of tissue is very necrotic, it's not healthy. But to get to the epileptic center, she had to hack through a bunch of healthy cortex. And that is discard tissue as far as the procedure is concerned, but perfectly healthy brain tissue. And she would give that to us. We would then were able to analyze that for single cell transcriptome data. And so the experiments are very similar to the mouse ones. We start with a piece of brain tissue, dissociate the cells. Here, Ben's group did some light immunopanning to enrich for neurons, but still have many different cell types in there. They were loaded into C1. We made the single-cell transcriptome libraries, sequenced them. And one of the things I like about doing these experiments with the microfluidics tools as opposed to FACS, we do both in the lab, depending on experimental requirements. But one of the nice things about doing the chips is you can take a picture of every single cell in the chip. So after we've done the transcriptome analysis, if there's funny results or something weird going on, we can go back and look at the images from the cell that gave rise to that transcriptome and ask, well, was there a doublet there, or a piece of debris or something else that might be confusing, or we can verify that they were in fact single cells and they were alive and so forth. And so in these experiments, we looked at nearly 500 single cells, mostly from adults. We also did some experiments with fetal brain, but the vast majority are from adult and of a variety of ages and genders. We sequenced each one through to several million transcripts. Oh, I'm sorry, that's cut off at the bottom. But basically, we could do a variety of QC estimates to show that we're getting reasonable results across these. There's not DNA contamination, the RNA is full length, so forth and so on. So at the end of the day, how do we analyze these? Well, we did a bunch of different ways. We were interested in using unbiased approaches like TSNE, which is shown here. And TSNE, of course, gives a bunch of clusters out, and then you can ask, well, are these clusters meaningful? Do they reflect anything about biology? There's no biology on the input. We're using the whole transcriptome and asking, and using we like to use the Karchenko distance to measure the distances between the transcriptomes. And we can ask if these clusters are biologically meaningful. And we've given them all names, and I'll tell you how we got to that in the next slide. Because the other way we analyzed this was to use our knowledge of neuroscience and use the fact that people have sorted mouse brain cells into, which are all believed to be sort of homolog cell types to the human, and they've done RNA-seq on them, and we're able to use that data to figure out what are the most highly differentially expressed genes in a given mouse cell type, and then ask, how do those homologs behave here? So we had a list of ones that differentiate the mice. We asked, do the human homologs differentiate the human cells? And it turns out they do in the different clusters and groups. And then you can ask, do the groups obtained by this biased approach that uses some knowledge of neuroscience agree at all with the ones we did with perfectly unbiased approach? And this is the agreement and the way we gave names to the different cell types. It's pretty good, 80 to 90% agreement, and it sort of all hangs together. So at this sort of course level, this gave us a lot of confidence in using these unbiased approaches to define different cell types. As you dig down into the different cell types, focusing on neurons, for example, that's an interesting one. We could build a minimum spanning tree and show that, in fact, there's different subtypes of neurons, both excitatory and inhibitory, and they're displaying all the markers you might think, and some new ones, and some interesting differences that aren't reflected in the mouse biology at all. And finally, let me give you a sort of taste of one thing that emerged from this that wouldn't have been possible without single-cell measurements, which is to answer this question about immune privilege in the human brain. There's been some controversy about whether the brain is immune privileged or not. Is MHC, in particular, major histocompatibility complex, expressed in brain cells and in neurons? And in most neurons, the answer is no. But in a small number, it's expressed at very high levels, equivalent to endothelial sort of control cells, which should be expressing it. And so you can see that even though the vast majority of neurons are not expressing HLA, there is a significant number that are expressing at high levels, and not just MHC, but other genes involved in the pathway. So it's sort of something that hangs together and is really the first, I think, convincing data that there is MHC expression in some subset of human neurons, and what the role is remains to be elucidated. So I'll stop here, and sort of summarize by saying I hope I convinced you that we have a sort of detailed understanding of systematic and stochastic errors associated both with gene expression measurement and genome sequence measurement at the single cell level. I didn't talk about the sperm measurement, but that was published several years ago. I did talk about this measurement of the clonal structure of tumors. I talked about the lung development, touched on the human brain work. And I didn't talk about this, but we've also done a little bit of work to do single cell epigenetics. That's sort of an early result there. There's more to come soon. So thank you, and happy to stop here.
Thanks, that was really great. I had a question. I wanted to follow up on one of your comments earlier in the cancer part of the talk about the futility of trying to computationally deconvolve the different clonal subtypes. And I can totally see the argument in the case if you just have a single bulk sample, but it seems less clear that it's futile if you have, say, multiple samples from a single cancer, that you might be able to infer clonal structure in that case. Do you think that's also futile?
Yeah, I mean, that'll depend a bit on the dynamics of how the clone frequencies change and if they do. You know, if it's a static thing from sample to sample, then you're stuck as the same as the one sample case. And so,
well, I was thinking of the case just like a single solid tumor in multiple sections.
Oh, sorry, you want to spatially section the tumor.
Sure.
Yes, so that's okay. That will give you some information about clonal differences. People have shown that there is variation on which is the dominant clone according to the geography within the tumor. So that's multiple measurements on the same tumor, and that can give you some information about clonal structure, but not purely looking at one sample.
Absolute beautiful work. My question is about the discreteness of these cell type distinctions. I mean, in your graph, you showed a little bit of these, what you called leakage, for example, in another cell type. You showed another one where it's sort of still making up its mind as to which direction it's going to go. So I'm actually quite surprised at how discrete it seems to be, rather than seeing much more continuity. And a related question was about the trees that you actually drew between your different clusters. So you draw this minimum spanning tree, but if you look at what I assume was a PCA plot or a MDA plot, that MDS plot, that basically grouped the different clusters together based on these single lines. I'm just curious, do you believe that these lines mean anything, or is it really truly clusters rather than a minimum spanning tree? So they're sort of related questions. Yeah.
So, I mean, the minimum spanning tree was just in the analysis of the neurons there, and so that's how we kind of parsed out that. And the other ones were done by TSNE and PCA in the places I indicated. And, this question of how well you can resolve different cell types, it depends on, in big part on the relative frequency of the populations. And finding a rare population and a dominant one is much more challenging computationally. If they're there at comparable amounts, it's much easier to deconvolve. And the question of identity itself is sort of a fundamental one, and these issues of plasticity, I think, are in many cases still unresolved. And part of the aim of the work is to get at that. I was able to show you very discrete ones in some of these cases because these are terminally differentiated, and they're different. But we've also done work with Marius Vernig looking at sort of induced neural differentiation from fibroblast to neuron, and as you're sort of doing expressed transcription factors, and these things are changing their identity, and you track that over time, you'll see the continuum there over time. Perfect. And so when you're forcing these things to reprogram, you can see that there's inertia in the transcriptome, and this vector is kind of slowly moving like this from one cell type to another, and it's hitting everything in between. In a very interesting way.
Is that a property of the induction, or is that a property of development itself? Like if you were to look at the developing brain?
Yet to be determined.
Okay,
yet to be
beautiful
determined.
Work. Thank you.